So today, common aseptic transfers, which we were just talking a little bit about. And then we're also going to do a streak plate method, so you'll also produce a plate. So aseptically, what does that mean? It means performing the technique so that we don't contaminate. Because as we already know from our very first lab, right, the air is a source of contamination. Our breath, right, our fingertips, our desktop. So we want to make sure to keep these cultures pure. Right? And so aseptic is just, an adjective. It describes a condition of being without contamination. So a mixed culture, as we said, is a media used for propagating in broth, slant, or plate form that supports the growth of two or more species. Right? So when we have a mixed, right, we have more than one. Typically, we want to work with pure cultures because that way we know what we're looking at. We're looking at a single species. So 4 of your cultures there actually are pure cultures. They should be just that organism on the label. We're going to learn an isolation technique, so if you end up with a mixed culture, like we purposely did for our broth, that you can separate them out, right? And the method that we're going to use today is the streak plate method. There are other methods out there, right, for isolating our organisms. This is the most widely used, especially when you have what's referred to as a heavy inoculum. You have a lot of bacteria in that tube. Colonies are visible growths on solid media, typically a plate because you have the most room for them to spread out. A single colony, if it arose from a single organism or more than one organism of the same species, it's said to be pure, right? But we don't always know when you're looking at that plate, right? The only reason you would maybe be able to know that those are hopefully pure cultures is because it says just one name on it, doesn't say a mix. And if you look at the morphologies, they're pretty much the same. But as you'll find out, right, as I've already mentioned earlier, some organisms are all the same color. Some of them grow exactly the same on plate media and slant media, on solid culture media. Right? It's not until you do microscopic examination and further testing that if you don't know what the organism is, that you may be able to figure out what it is. And that's something we're going to work on after midterm. So you guys are going to learn all the techniques now, all the staining and stuff you'll notice coming up, and how to manipulate and move the organisms from one form of media to another aseptically without contamination. And then you'll need to do that to figure out what your unknown organism is when you are given it after midterm. So a colony forming unit is a term that we use to define the cell or the grouping that you see on the plate. And typically when we use plate media, we're trying to spread out the organism. So a single cell or just a few cells of the same species are together. So when they grow up into a colony, and we can count those colonies, and again, we can hopefully think of them as being pure, as being a single organism. Not always the case. Kind of makes sense to you guys? Right? So this is terminology we're going to use from this point forward. Right? So if any of it's confusing, ask me. I'll try and say it a different way. Notice I didn't read the slides, right? Because they say it a different way. Right? So you don't have to know the exact definition. You have to have an understanding of these terms. Does that make sense to you guys? All right. So one of them was even on your quiz today, was the bonus, right? And I have your quizzes from last week, but last night I was like, what am I supposed to do for lab? I'm like, I know I'm supposed to look at my lecture, which I didn't do, but I was like, I can wing it. And then I was like, what else am I supposed to do? I was supposed to enter you guys' grades. The good news is while I was sitting at the hospital waiting on Monday, I got them all graded. They're like, oh, the technician's not going to be in for another half hour. So it's okay, I always bring work with me. I'm not stupid. Because you people never move quickly. Or you do stupid stuff like not put me on the schedule. So, a pure culture, we said, is one species. right? So we're starting out with pure cultures for the most part today for our aseptic transfers. So we have different types of media, so you guys get practice dealing with the different types. Pretty much from now on, though, we'll deal with mostly slants, because slants give us a nice solid surface, and they're in a tube, so we can store a whole bunch of them very easily in a small space, right? So for storage reasons, these work much better. Some of the organisms we work with are motile, which is great. They could swim around in a broth, right? And hence why that one that's really cloudy all throughout, there's a motile organism in there. He's swimming all around. And there's a non-motile, right, in there as well. Hence the clump at the bottom. Or dead ones that have fallen to the bottom too. So they would be happiest if we had what's referred to as a shaking, or one of the labs I worked in, literally you opened up the incubator and there was this big wheel that had all these holes in it for the tubes, and it spun, right? And so this moved the media around for those ones that weren't motile, so they weren't basically, you know, stuck in their own swill, their own waste all the time. They were getting fresh nutrients for part of the time. So we don't have that type of equipment here, right? So we don't usually utilize broth cultures very often because the microorganisms aren't very happy, most of them, in that type of situation. Where if they're stuck on a flat surface, even that slant, it's much easier to acquire nutrients, especially for a non-motile organism. You can't move. Plates are great too, right? Very large surface area. The problem with them is that whole lid thing and how big they are, right? And the separation of lid from top and, you know, potentially becoming contaminated. We're getting those lids off the tubes is less likely to happen, especially if you grab the tube by the tube like you're supposed to instead of the cap. Make sense? Okay. So today, right, we have sterile media that I have set aside for you guys warmed up in the incubator. And so it has no organisms in it, right? Sterile means no organisms at all. So from your guys' quiz last week, you guys remember what we called our plates that we didn't expose that had sterile media? What were they for our experiment? They were our negative control. Yeah, remember that? It's important. I was like, it's difficult grading some of those quizzes. Okay. So, and we'll talk about some of that stuff in the quizzes, but I'm not going to hand them back today. I'll hand them back after Mardi Gras with this one, because I just didn't enter your grades yet, so I can't hand them back. So, as I said, you know, you can transfer from any type of media, right? But depending on the type of media, there's a procedure, a way in which you need to inoculate the different sources. So we can go from a broth to a broth, a slant to a slant, a plate to a broth, which is one of the things that we're going to do today. So that initial source, that pure culture, is typically referred to as the inoculum, right? And you may have a lot of inoculum to start with, right? Or you may have a really small amount. So we would say, oh, heavy or light, a lot or a little. Inoculation is what you're going to do. You're going to take from your pure cultures and you're going to inoculate, right, add bacteria to the sterile media. Sometimes aseptic technique is referred to as sterile technique. Because again, we don't want to contaminate. We want it to be just the organisms we're working with. I don't like using the word sterile because sterile to me means no microorganisms at all, right? So when you say sterile technique, it'd be like, okay, seems counter opposite. But you'll sometimes hear it referred to as that. I'll always say aseptic. So the food sources, right? So in that media is food. In addition to agar that we talked about a while ago, right? This is the solidifying agent. This is why it's solid in the plate, why it's solid in our slant tubes. And then we have beef extract, right? So just like us, they like hamburgers, basically. right? And peptone is a partially digested protein source. So it makes it easier for them to grow faster. So it's very, you know, diverse food source. Most of them, especially pathogenic ones, like to eat it. So it's, believe it or not, not all that different from us. So nothing is digested in the beef extract. So tryptase soy agar is another one that we commonly use. So what tryptone is a digestion of casein, which is a milk protein. It's actually the protein that makes your milk white. And then the soytone is a digestion of soybean. And so they use pancreatic enzymes. Like if you see the word trypsin, it's referring to the enzyme that they're using from the pancreas to digest proteins. So these are really rich sources. They support a wide range of growth. Lots of organisms like to eat them. No problem in getting them to grow. So what we typically use to just propagate, get them to grow. Well-defined medias, right? We know the exact chemical composition. So what I usually refer to these as is like the Nutrisystem diet. So of course, that type of media is very expensive. We only have one that kind of falls into this category that we'll use later, second half of the semester. And the reason why we use it is a specialized media for running a particular test. And it just happens to be defined where everything is measured out in it. And we'll use even more specialized medias that help select for the growth of organisms and help us differentiate growth, right? Those types of special media we're going to use the second half of the semester. The first half we're mostly just going to be utilizing standard nutrient agar, nutrient broth, tryptase soy, agar, and broth. We did see some specialized media. Remember the blood agar plates that I brought out in the beginning? Right, so they were specialized in that they had blood in them, red blood cells, whole red blood cells. They were affected by the growth, and they actually even had antibiotics in it too, so that only gram-positive organisms would grow. So why do we do this? Well, think about this. We just basically, when we stick them in these tubes, we basically just stuck them in a fallout shelter with a whole bunch of MREs, and the toilet just broke. That's kind of a crappy environment, huh? I don't know about you, but I'm not signing up. You're not sticking me in a room with a whole bunch of MREs and then turning off my toilet. But that's what we do to microorganisms all the time. We give them a whole bunch of food, we stick them in one place, and we don't let them get rid of waste. They got to live in their own swill. So as you can imagine, over time, a lot of them start dying and stop making babies because it is not the greatest environment for making babies. So if we want to keep them happy and growing, what do we got to keep doing? We got to keep moving them from one fallout shelter to the next. So to revive those stalks, to make sure that they're actively growing and happy. So we use a loop or the tip of a serological pipette if you're using a frozen stalk, which you guys don't do, but you may in your future careers, who knows. So a differential or selective tests, again, we're doing one step further. We're actually helping identify different properties and select for different groups of organisms. But same thing, we're going to inoculate it, right? We're going to put organisms in that environment and see what happens. Sometimes we may need a needle, right? And so in this case, right, this long, right, nothing at the end. You guys, today we're going to be working with the loops. So at the end of the metal, right, is a little loop which helps you pick up the organisms. It's a little bit more surface area than just the tip of a needle. These are my crappy old ones, which I was going to cut off and twine, but I keep forgetting to bring my wire cutters in. But these are the good ones, so we should have enough for everybody. The ones that are in the beaker. Differential, a wide range of organisms will grow, but we're able to differentiate based on their growth, right? So if I had remembered to take our blood agar plates out of the incubator, you actually would have seen kind of a greenish tinge to them because of the damage they cause to the red blood cells. As time goes by, you don't see that. But we'll see it again. Selective, as I said, will select for particular organisms. So usually we have something toxic in the media. In the case of the blood agar ones, it's antibiotics, right, which are going to kill the gram negatives and allow us to grow the gram positives. So why is it important that we use proper aseptic technique? Right? Reliability of our experiment, right? Keep the environment out. We want to keep it pure. We want to make sure it's just that one organism we're studying. And we also don't want the media doesn't become contaminated with additional species.
Excuse me.
Uh-huh. Oh, okay. Put it in the closet. Yeah. Safety, right? We don't want to contaminate our environment. Did we have a spill over there earlier? Is that what happened? Did we spray Lysol? I think I saw you grab the Lysol. Okay. So again, with these cultures, if you spill it, you're spilling microorganisms, right? We need to disinfect when that happens, right? And so we're going to use the Lysol bottles to start and end when we're working, and then if we have a spill, definitely make sure that we add Lysol to it. So make sure our possessions or our body doesn't encounter any unwanted organisms, which is why, especially today, right, jackets hanging on back of chairs, all that stuff needs to go in the closet, right, because you don't want to spill on it. Make sure that no one else in the laboratory becomes contaminated, right, so if she hadn't wiped that up, right, someone else might not know it's there and picked up a large number of microorganisms. So it's important today, too, we're going to be wearing our aprons, we're going to be wearing our goggles, we're going to be wearing gloves, that, you know, if we need to leave the room to go to the bathroom, get a drink of water or something like that, take your gloves off, take your apron off, and do what? Wash your hands. Even though you're wearing gloves, wash your hands, right? So the other technique we're going to do is streak plate. So you could have multiple organisms. Today we're working with a broth, but they could be your slant might have got contaminated and you just separate out the organisms, or you might have a plate. As you can see here, you can definitely tell there's more than one type of organism because there's different growth patterns here, right? There's a lot of different colors. On this one, not so much, but you can see this pattern, which unfortunately we weren't able to do on those plates. I wonder if I still have one of my stashed plates. Yeah. So this one's kind of dried out, but yet still liquidy. And so you can see, right, you're going to put the organism and you're going to pick up a lot of organisms. So you'll end up with a lot of growth. And then as you spread it out in this technique, you're going to eventually get to where you've got single organisms, hopefully growing into a colony. Hopefully it's pure. Dried out, but still got quite a bit of moisture in it for this skin though. Usually the starting plates that you guys have are streak plates. But remember I said about bacteria not always cooperating? That's an example of non-cooperation, right? Because we're lucky we have growth on those plates. I don't think any of them ended up looking like a streak plate like this one. So I'm gonna clean off the outside of this one. So this is not a pathogen. This is not Klebsiella. You can pass that around to see what we hope our plates will look like next week. You can see that media in there is like dried up, about ready to be done. Pass this all the way around, sir. So a single strink of inoculum is light. For example, when you guys took from the desktop, right? We just used the swab and ran it across the plate. And that was enough because we didn't have a large number of organisms to get all those different growths we saw on our plates last week. But if you have a heavy inoculum, which is what we're starting with today, a lot of microorganisms, or picking a colony, that's a lot of organisms. That's like 1 billion bacteria in one of those colonies. You'll use this streak plate method that we're going to teach you guys how to do today. So there are different ways, right? Beginning scientists typically draw them out, quadrants, sections on the plate to help keep track of where they've been and where they're going. So I have an example of that on the handout that you guys have today. And we write on the bottom of our plates, and I'm going to show you how to do that. And that can help us orient around the plate as to where we're going and where we've been. So in the first quadrant, you would apply your organisms, with the loop, and it's invisible coloring, right? At this point, you're not going to see them. Teeny tiny, right? When we come back, hopefully we see that large amount of growth. But you'll notice that this is that first quadrant, but it kind of crosses into what's going to be our second one. So then when we start in this quadrant, we're going to be picking up organisms from the first one and spreading it into the second quadrant, and then even partially into the third. In between each time, we're going to do what to the loop? We're going to flame it. What does that do? It kills the organisms. So here, right, we're spreading them out as best we can, and you want to cover every single little inch you can, right, as best you can, and fill up that whole quadrant. And then you're going to flame your loop, and then now you're just going to pick up, by crossing into this one a few times, organisms that you already put on the plate. So hopefully you're picking up less than, of course, your initial inoculum. So then you're spreading them out. So hopefully you're getting them even more spread out. It's like dilution, right? Flame our loop. Pick up from the second into the third. Flame our loop. Pick up from the third into the fourth. Usually by the third or fourth quadrant, you're going to get isolation. Klebsiella was just being difficult, right? So even though she probably had a pretty heavy inoculum, a lot of the organisms probably weren't happy, and so therefore a lot of them didn't grow. So she got isolation in like quadrant 1, right, on some of those plates that I gave you. I always kind of equate this when you go to those fancy restaurants and they give you like 1 pat of butter, and you're trying to, when I ate bread, I try to spread it out over the whole surface of the bread so each bite had butter. Yeah, with those little pats, it's like scraping it thin, right? So imagine, trying to spread it out as best you can. So depending, sometimes you'll start out with a swab and then a loop. We're going to do all loop. So that kind of had angled. I kind of draw mine straight. I'm not very good at angling, right? It doesn't really matter. You just got to divide up the plate, right, and be able to follow your pattern. So I call this my goalpost one, right? It looks like a goalpost. Right? So I say, draw your goalpost. Get yourself oriented. But same principle, right? And this is another variation of that, where they don't have the lines intersecting, right? And but you've got to cross the lines, right, to be able to get into the different quadrants. And I typically, when I like to do this one, when I go from 3 to 4, instead of a continuous streak, I pick my loop up and I do single streaks. So I drag just from right here over, just from right here, pick it up, drag over, all the way down the whole surface of my 4th quadrant, right? But you could pick up a little bit from here and just streak on down, you know. Just make sure you make a note of what technique you utilized, right, or what variation of a technique. A good scientist always writes down what they do. What happens if you make a mistake? You write it down, right? I'm not going to give you extra media today. You're learning. You're going to make mistakes, right? You just need to record them. So, Peter Hickman worked in a research lab. Again, you don't want to be using like a gazillion plates. He would literally divide up his plate into several sections and do little tiny streak sections using sterile toothpicks, right? So he'd literally put one little line, right, and then one little line and one little line, and then streak out the rest. So that would be one of his samples, right? And then he would do the same thing for each one of those wheels around that plate. And so he could get how many samples on there? 8 different samples on one plate. Doing it at micro scale. So we've already kind of mentioned about what the organisms look like when they grow. So today we saw mostly creamy white, right? But later on we'll see even kind of chalky, dry looking white cultures. Others like this one, right? You're kind of looking at it, go, I don't see anything, right? Kind of translucent. There'll be some that'll be cloudy all throughout, especially motile organisms. There'll be organisms that require air, that are strict aerobes, and they'll only grow at the top of that tube, because again, if we're not mixing that tube, where's the oxygen? At the top, right? So these are the ones we have for today. Thankfully, they all grew, right? And we only did one mixed broth today. In the past, we've done different ones. Now, as I said, I don't expect you guys to know this terminology or learn this terminology. We're not going to take out dissecting scopes and look at the colonies to this level of detail, but you definitely can see that irregular edge, hopefully, to the Klebsiella, right? And then, you know, if we were to really look, right, you might even see these different raised platforms or kind of convexed. And actually, I think Micrococcus luteus, see how it looks like a hill? Do you guys see that? Like a mound? So when describing, and this one's actually on blood agar, they're white, right? They're raised, they're circular, they'll be smooth for Staphylococcus epididymis. For Klebsiella pneumonia, again, white, they're raised, right? They tend to be circular and smooth. Sometimes they'll even look mucoidal, right? Mucousy and shiny. And then, of course, Micrococcus luteus is yellow, right? The term would be convex. They're circular, they're smooth. This media happens to be brain heart infused. And then, of course, this is another blood agar. And even notice the difference in time here. So this is Bacillus subtilis, a different bacillus than what we're using today. But it has a very unique growth pattern, right? This is 24 hours out, and then look how it looks, right? You have this spreading edged, right? You have this raised region. It's very rough and dry looking 2 days later, 48 hours on blood agar. So these cultures that you guys are working with, Micrococcus luteus is a slow grower, so those were actually inoculated on Monday last week. And the rest of them, I think, were inoculated on Wednesday last week. So they're pretty old cultures. So these guys are like, yeah, I'm ready for a new fallout shelter. Please rescue me. Right? They're not happy. So as I said, this is the plan. Do I need to leave that up for now, or are you guys good? Do you guys all get it written down on your little section, the plan? Okay. I need to write up my plan. I might just leave it up. Okay. So what we're going to do next, I'm going to actually put my lab coat on. You guys can get goggles. I feel pink today. Right, we have the spray, or you can use the wipes. Remember the important thing with these guys, open up these vents, right, so we don't clog up. And you have paper towels on your lab bench too, well, as I got some up here. So, get you goggles, get you an apron, grab you a pair of gloves. I'll wear my new one today since I'm not using the same. The other one looks like I've been in a color war. Oh, did I hit stop recording? Let me do that.